More than 25% of cancer deaths are caused by chemotherapy-related toxicity and not the disease. The same drug that could save a patient's life could kill another patient. The reason is that there's no two bodies that are the same. Thus, there's no two cancers that are the same. Imagine a future where instead of testing directly on a patient, we can test on a living model of the patient's body on microchips, where we can determine exactly which drug is perfect and ideal for this patient, and what's the precise dosage that this patient's body could live with. Even drug discovery is not optimal. We don't have actually enough drugs to do so. Currently, it takes $2.6 billion and 12 years to develop one successful drug, because 90% of the drugs fail at the late and very expensive clinical trial stages. The reason is that preclinical results do not determine clinical failures. So with the tests that we do preclinically do not translate to the clinical results. Imagine a future where instead of having like the number of drugs that we have today, each year we develop hundreds of more drugs that could benefit for precision and personalized medicine. Actually, to understand it better, we need to understand exactly what's happening at the preclinical results and why they are not accurate enough. So lab-grown tissues used in preclinical drug testing do not replicate the tissues that are grown inside our bodies. And the reason is that cell behavior is strongly influenced by two things: the 3D tissue structure of the tissues, which is the arrangement of cells within a tissue. So cells are beside each other and they communicate with each other. And the tissue microenvironment, which is the dynamic interaction of the cellular and the surrounding non-cellular components. So all the microenvironment is important for the cells. Conventional solutions do not preserve neither the 3D tissue structure nor the tissue microenvironment. To understand it better, I'll give you like a brief of what they do currently. So currently, they test cells on 2D plates or 2D surfaces, which not just lack the mimicry for the tissue, the resemblance for the real tissue into the 3D tissue structure and the tissue microenvironment, but also it's just one tissue type. So it lacks the full body communication. Our body is not just one tissue. It's a magnitude of different organs that are connected together, and they talk to each other. What about animals? So why animals are not good? Well, basically, they're just like not humans. So they're not accurate. So we actually focus on something called ductal tissues. Ducts are found all over our bodies. They are found inside of our lungs. We have the bronchioles. They are found in our kidneys, in our liver, and in most of our organs. And they play a functional role in the functionality of most of our organs. Actually, like 85% of cancer cases occur in a way or another in ductal epithelial tissues. And those tissues are not accurately modeled today in the lab. The solution is called Organon chips. So what's Organon chip? An Organon chip is a small plastic chip where we can insert cells inside of this chip, living cells, and those cells could attach to the surfaces that are designed inside of this chip to form 3D tissues that resemble the tissues that are found inside of our bodies. We can use those organs, those microorgans, to test drugs and to better study the effect of the drugs before testing on humans. Great. What about ductal tissues? There's a problem because ductal tissues are one of many tissues that are a bit more complicated. We have this 3D structure that's not as simple. So to solve it, we actually have cylindrical ducts, cylindrical scaffolding systems. So what's a scaffold? A scaffold is a thin surface where we can insert cells inside of the scaffolds, and the cells could attach to the surfaces that are designed inside of the scaffold. Great. But this scaffold is ultra thin. It's 7 micrometers in thickness, so it's thinner than a cell. And it's porous, so when we insert cells, the cells won't slip to the other side, but they have the opportunity to squeeze and pass to the other side if there's a signal that allows them to do so. We can insert cells inside of that scaffold and from the other side of the scaffold, so to replicate the full tissue that we aim for. On the right, you can see a chip that we created in the lab, and below we can see the living tissue that we grew inside of this chip. That's a simple cylindrical duct, but things could be much more complicated than that. So the good thing about scaffolding systems is that it's personalized, it's customizable. We can create any shape, any pore size, or like holes inside of the walls. We can create like a magnitude of different ducts, of different structures, of different surfaces. So we can have like multiple solutions for different problems. For example, we can have a duct and surrounded by multiple tissue types to study, for example, the metastasis of cancer, or like why some cancers metastasize to a specific organ and not another organ. We can also like put two different ducts together to model, for example, more complicated tissues, such as, for example, the liver, where we have like a bile duct and a hepatic duct. And we can study, if we have a tumor inside of it, we can study also like the vascularization that feeds the cancers. Great. So we have a tool that can allow us to create like more accurate tissues. But where to use those tissues? We can use it to study kidney and liver toxicity. So our kidney is a filter of our body. It filters the blood. And if this filtrance goes to urine, but before passing to the urine, it passes through ducts in the kidney that we call the renal proximal tubules. Those tubes are simple tubes. They reabsorb the good stuff that we need in our body. So for example, glucose. So because they have this high absorption rate, if there's a toxicant in the blood that's filtered, the first cells to die are those. So we can study kidney toxicity by measuring the amount of cells that are dying using inside of these models. And we can also model like more complex models such as the liver, where we have like multiple ducts with different types, with different tissue types. And we can study like the hepatocytes toxicity, which is found in the stroma. We can also model cancer. So we can grow healthy breast, prostate, or lung tissues, lung ducts. And we can grow a solid tumor inside of the duct. And we can study the effect of a drug on the efficacy. Is it killing the cancer or not? Is it stopping the growth of this cancer or not? We can also study something called the blood-brain barrier. Not all the drugs reach the brain. There is something called the barrier that prevents many drugs to reach the brain. So we can grow a tumor inside of the brain, and we can pass the drug inside of the duct, and we can see if the tumor in the brain is being killed or not. We can have something even more complicated. We can have a blood vessel and a breast duct, for example, and we can study metastasis, the vascularization from the vessel to the duct. That's great. So we have an accurate model for a single organ. But is that enough? Well, unfortunately, it's not enough, because our body is much more complicated than that. So our body contains multiple organs that are interconnected together through immune systems. We have hormones, we have biochemicals, we have lots of things. All of those organs talk to each other. And the effect of the drugs on a specific organ is dependent on the magnitude of different things. Thus comes the human-on-chip operating system. So it's a bigger device that consists of two different parts. The first part is the human-on-chip, which is a chip that contains multiple organs for a kidney, liver, heart, brain, lung, the major organs inside of our bodies. And they are connected together using microfluidic channels. Okay, that's a static thing. We can insert this inside of a bigger machine called the operating system. And this operating system can automate all the experiments and can model the dynamics that we have inside of our bodies to create a replica of not just the tissues and the organs, but also the functionality of our body on the chip. Okay, is that enough? Well, it's not. Because what we can do using this operating system is we can automate an experiment. We can measure everything in real time. We can connect different organs together, and we can model the body's functionalities. But what we can do is analyze and optimize in real time, because those are dependent on the user input. So there's users, and there's limited numbers of users and limited time, and there's millions of parameters that we have. That's where AI comes, above the operating system. So what the artificial intelligence does, the operating system can measure and can manipulate the system. By just adding a learning step, we can automate the analysis and the optimization in real time, and we can get like a number of information that's limitless. We can know a lot about the drug, about the disease, about the effect on the tissue, about the effect inside of the cells. We can know like about the dosage, about multiple things that it's impossible to know before. Okay, great. So where does the Organon chip fit in everything? So currently, Organon chips can replace animal testing. It's a new bill actually that's issued so that FDA is legal to get it without animal testing, but to replace it with in silico and Organon chip testing.
But with adding just this organ-on-chip step, what we can do is that we can early terminate the drugs, some of the drugs, before entering clinical trials, and we can cut the cost by 50%. We can improve the formulation of better drugs. And most importantly, we can prevent some good drugs from being rejected at the animal trials and getting to the market. For example, aspirin. Aspirin was invented more than 100 years ago. Aspirin causes toxicity in mice. So imagine if aspirin is discovered today, it won't reach the market. So we can also predict those false negatives. So what's the future of this technology? What we can do also, we can do something called population on chip, not just a random human on chip. You can model children, elderly people, people from different backgrounds, having different conditions. So that, for example, there's lots of drugs that are being developed for children while not being tested on children, and they reach the market. And even further than that, we can do something called precision medicine, precision and personalized medicine. We can have a model for each one of us, a model for me on a chip, for each one of you on a chip. We can model the full body. So when we get like a disease, we can just do the testing on those chips, and we can determine exactly which drug is optimal for us and with which dosage. So we can de-risk the deaths that are preventable. That's just a glimpse of the future of precision and personalized medicine, the future of drug discovery. The future is limitless, and it's up to your imagination. Thank you.